Good morning and good evening to all of our attendees joining from Korea and also US. My name is Boyoung Cha, and I'm the Senior Program Coordinator of the Johns Hopkins Global Biotechnology Innovation Center, which is called JHBIC. So my pleasure to welcome to all the March session of the JHBIC webinar series, proudly hosted by our center. So today's topic is particularly exciting as it dives deep into the cutting-edge frontier of the precision medicines and featuring innovations in spatial omics and the SLEX, the spatially resolved laser-activated cell sorting technology. So we are honored to introduce our distinguished speaker, Dr. Amos Chung-won Lee, the CEO of Meteor Biotech and a leading scientist in spatial genomics and then translational medicine. So Dr. Lee holds a PhD in bioengineering from Seoul National University and a BS from UC Berkeley. He has honored numerous high-impact publications, including recent work in Nature Reviews Cancer and molecular cancer and genomic biology, etc. And his innovations have earned multiple prestigious awards, including the Pogo Young Scientist Award and the Organoid Young Scientist Award. So today, he will be presenting on advancing precision medicine with the flex and the spatial omics. And it is my great pleasure to welcome Dr. Lee to begin today's presentation. So please welcome Dr. Lee.
Thank you, Dr. Cha and Professor Kim for inviting me to this honorable seminar. And it is also my great pleasure to present to you about our research regarding this technology that we developed, technology that is very proprietary to our company. So let me share my screen. Yes, so I'll begin my seminar. So my presentation today will be comprised of the introduction of the technology, the company, and then I will have several examples of published works, and non-published works. So please feel free to share the published works, but please don't share the unpublished data that is marked with unpublished for sharing. So today, I entitled my presentation as Enabling Diagnostic and Therapeutic Target Discovery Through Spatial Cell Sorting with Multiomics, and we call this technology as Spatially Resolved Laser Activated Cell Sorting Technology, SLACS in short. So we named this after FACS technology, which is a short for fluorescence activated cell sorting technology. But instead of it being fluorescence activated, we call it SLACS technology because it's laser activated and it preserves the spatial information, so it's spatially resolved. So we sort cells basically with preserved spatial information, and we perform DNA sequencing, RNA sequencing, or proteomics using mass spec so that we could produce spatial DNA sequencing data, spatial RNA sequencing data, and spatial proteomics data for discovering very specific and significant targets, whether it be scientific or clinical, so that we could understand the mechanism behind the biology or the disease of or in certain biological systems, and define this very specific targets, molecular targets, so that it could be translated to a therapeutics or diagnostic marker. So we are aiming to develop this spatial cell sorting technology with a very high clinically or scientifically relevant discovery. So this I'll start with my introduction of a history of landmark bioresearch devices. So if you look at the field of bioresearch devices, you can see certain pattern of academic development of certain technique, commercialization, and spawn of a next generation techniques. So it all starts with a very basic question, such as how are we genetically encoded? To answer this question, the U.S. government has launched the Human Genome Project which aim to sequence one person's genome for 10 years using around a 1 billion US dollars, so that we could map out the whole genome of a person. So that started from 2000, around 2000, and it lasted for a decade. And you can see that in 2008, the Nature Methods has selected generation sequencing as the method of the year. So because the engineers and technicians and scientists have developed this technology called NGS, next generation sequencing, to answer this question of how are we encoded very fast and with lower expenses. And this was commercialized by a representative company called Illumina, and it's now called the era of $100 genome, where one person can be sequenced, the genome of one person can be sequenced with just $100, and it could be sequenced in one day. So this technological improvement not only comes from the academic development of these technologies, but it also has to be supported by commercialization and productization of these technology to have a real world impact of making this technology available and very useful for engineers, scientists, and clinicians. So Illumina now has around a 30 billion US dollar market cap, which is way higher than the 1 billion US dollar that the US government has input for sequencing one person's genome. So it's this cycle of academic development and translation, commercialization, and productization, and R&D development in industrial level. This whole cycle of development of bioresearch devices has caused very huge innovation in the field of bioresearch, where people could sequence several people's genome at very in a very cheap and very fast manner. And the next question, now that we know how we are encoded, was to figure out what cells comprise certain systems of biology, such as if you have a brain or if you have a tumor, what cells would comprise these systems so that different, very heterogeneous cell populations would interact with each other and cause this state or disease state or normal state of certain biological systems. So the efforts to do this um, was launched by the Chan Zuckerberg initiative called Human Cell Atlas. And this was also a very expensive and very laborious procedure to sequence one cell at a time. But in 2013, single cell sequencing is named as a method of the year by Nature Methods, and this was successfully commercialized by a company called 10x Genomics. And currently, single cell sequencing is getting cheaper and cheaper with the innovation given by 10x Genomics, and the company itself also has 4.3 billion US dollar market cap. And where if you think about how the Human Cell Atlas has put in around 1 billion US dollar as a investment, um, this this rise of new technology was a accelerated by innovations that are made from this industry. And now that we know how we are encoded and what single cell comprise us, the next step, the next question in biology was to put that cells in a biological circuitry, or place them in in the spatial dimension, so that we know how this biocircuits work with these different cells, and how they produce certain structures, and how they function in terms of spatial information. So the next question was, how are these cells located and positioned so that they would have this certain type of spatial information. So the spatial omics was first named the method of the year in 2020, and 2000 recently in 2024 by the Nature Methods. And there are many, many, many spatial spatial omics companies, such as 10x, Akoya, Acuity, Ultiview, Curio, and so on. There's very many, many, many spatial omics technologies where different companies have their own proprietary technology to address this question of how the cells are located. And there are still trials and efforts to bring this academic innovation to the field of industry field, so that it could be used as a innovation for innovating this area of biology. So, what we think, why this spatial biology tech race is happening right now, especially around 30 companies were um, st started in recent 5 years, and there are many, many active acquisition and merge of spatial technology, and major biotech and small biotech companies are racing with their own spatial tech. But because we think this is because these major bio companies and customers in biological area want deeper insights into biology, basically, or diseases, so that they could find the targets, so that they could be translated as a multi-million dollar or multi-billion dollar drugs, therapeutic drugs, or a very good diagnostic tool, so that the patients could have a chance for receiving personalized or precision therapy or diagnostics in ultimate goal. Um, so I was looking into a talk given by a speech given by Larry Ellison, the CEO of Oracle, in January of this year, um, as they were launching the Stargate project. Um, Ellison said something like this: AI will design mRNA vaccines for every individual person against cancer, and they announced a plan to build 20 data centers, each 500,000 square feet, to power AI for analyzing health records and assist doctors for improving patient care. And I think this data centers would contain very different uh, aspects of health data, which would contain health records as well as genomic data and other omics data, so that it could aid the patient's the understanding of the diseases, so that we would have a data set for the AI to learn from. And this AI would ultimately provide a cure or diagnostic solution for these different disease states for for every diseases and every individual person ultimately. So if you think about how this would work, we think that the solution, like spatial omics solution, and a very large wave of active activity of the spatial omics space in biology area, would ultimately cause the major companies and different customers, different users of spatial biology to discover different targets for curing the diseases and then diagnosing the diseases, so that something like Larry Ellison has told can become true and become realistically a realistic solution for people. So he says something like producing 48-hour producing mRNA vaccine cure for tailored mRNA vaccine cure for cancer patients in 48 hours. But using our technology, we are envisioning so that we are to some spatial omic data from our technology can produce something very valuable to mine from, so that these AI can mine a very in a very effective manner, so that they could produce a very high quality biomarker data. They could even make a whole new industry, whole new field of industry, so that it could arise a new markets and provide cures and provide diagnostic solutions to people worldwide. I can give you one example from diagnostic biomarker and one example from therapeutic biomarker. One example is a breast cancer genetic test that was developed by a professor in Yonsei University in Korea called Oncotype DX. It has made a very good diagnostic solution for breast cancer patients, and it is widely used around worldwide, and many patients are benefiting from this solution, where this is just a solution of different biomarkers, different markers from breast cancer tumor mass, so that these patients could receive tailored or precise medical solutions when they are needed. And also a very famous example in therapeutic biomarker, immune checkpoint inhibitor um, targets PD-1 PD-L1 interaction, and it has also, uh, around 1,000 billion US dollar market worldwide in pharma. So, and many people are benefiting and surviving from this therapeutic biomarker. Um, so this development of or discovery of diagnostic or therapeutic biomarker, we say, is very huge and meteoric, so that it would have a very good, very huge impact to the people, and it has a very great value to accomplish. Um, but traditionally, it was really difficult, and it takes a lot of money, lot of time to discover a certain biomarker from certain diseases. Because, for example, if you take cancer, you have a late stage patient samples, normal patient samples, early stage patient samples, and you have a tons of samples to look at, 100 in minimum. You would have to look into different structures of tumor, different cellular phenotypes, different H&E pathological information that you could acquire from these different H&E sections of different tumor sections, and with different patients with different stages. And you have to look into different cells that are inside these tissues. There are a huge amount of cells in a tissue section, and even more information in DNA and RNA and proteins in even single cell. So if you multiply these information from the cohorts, the cells, and genetic molecules that are inside these cells, there are too much information that we can mine from. So it's almost like looking for a needle in a haystack, and you would have to do this very fast and very inefficiently until you had a spatial omics solution. So people are adapting AI or different solutions to mine um, uh, targets from this ocean of information. But our solution is to just look at the spatial information and look into the cells that are relevant to the diseases and target that cell and dive deep into that cell and look into the deepest information such as DNA, RNA, and protein, or multi-omic approach to that looking into that cell so that we could find the target of that cell very efficiently and very in cost-effective manner. So this by spatial information, I mean this is a figure that we published in 2024. Um, you have the proximity of the cells, the relevant cells, the cellular composition of the tissue, cellular morphology, and tumor structure or any other structure from different biological systems such as uh, the brain, the organoid, and even brain on chip or organ on chip. So any biological systems would have these structures, proximity, cellular composition, and morphology, which are all important in understanding the biological function or biology behind any type of diseases. And so to find these targets, people had developed different spatial omics technology. And basically, spatial omics technology is connecting this spatial information to the omics information. So our the motto of our company is what you see is what you get. So if you have a what you see part such as H&E staining, immunofluorescence staining, fish staining, or multiplex IHC staining, you have that image information that you see with your eyes, and you have a what you get information, which could be um, different omics data such as DNA sequencing data, genomics data, epigenomics data, transcriptomics data, proteomics data, or even metabolomics or lipidomics data that you could acquire from these different cells. So if you can connect that spatial or pathological information to the omics part, you can connect that image feature of different biological systems and biological functions to the molecular targets so that this could be translated to a very useful drug targets or diagnostic markers. And companies like 10x Genomics, NanoString, Vizgen, Akoya, Lunaphore are mostly focused on connecting the H&E staining part of the what you see part and gene expression typing based transcriptomics. So it's only for connecting the RNA sequencing and the spatial information at this time. And we sought to make our technology a very useful technology that the researchers, so the users can do mix and match of every what you see part of the staining technologies or spatial technologies and every aspect of what you get technologies like DNA sequencing, mass spec, and any type of omics technology that are available out there. So we again renamed this technology as SLEX technology, which is a short for spatially resolved laser activated cell sorting technology, which uses infrared pulse laser. So it just uses a very thermal energy based ablation of the cells of interest as it is targeted with uh, a very weak power infrared pulse laser, and the pulse is just a few nanoseconds, so it would have a very short, a very minor effect to the cells. When we measured the cells, the effect to the cells, the temperature of the cells went up by a few degrees, and we could even sort live cells, although very it has a low yield. We can even isolate live cells and try to culture them so that they could form colonies also. So it has a very minor effects to the counterpart technologies. So most spatial omics technology utilize ultraviolet laser, which could cause a thymine dimer formation, nucleic acid denaturation, fragmentation, or it could also cause effects to amino acids, so that the results from the omics techniques could cause a very different artifacts and different. It could be also very detrimental to the genetic components inside the cells. But um, on the other hand, the infrared laser has a very minor effect, and we have a very thin layer of transparent glass-like physical the glass-like structure of layer, which we call discharging layer, which vaporizes from solid state to gaseous state if it meets the infrared pulse laser, and so that the thermal energy of infrared laser will be converted to mechanical energy, pushing out the cells of interest or parts of the tissue so that they could be retrieved in a 96 well PCR2 format. So it has a speed of 1 target per second speed, so it's really fast, and it could be preserve different image information, and so that we could uh, produce omics data and map these omics data back to the spatial data, so that we would have that connection between the spatial or pathological information to the omics information. When we do this kind of spatial omics technology. So what we've tried is to basically um, produce different research from using different indications, different diseases, to connect different parts of the what you see part and different part with the what you get part, so that we can have that mix and match of different spatial omic data, so that the users could see and look into these data, so that they can find the drug targets and diagnostic targets. And we've tried this with FFPE samples, fresh frozen samples, um, bone marrow smears, organoid sections, or even IHC stained tissues, multiplex IHC stained tissues, fish stained tissues, fish stained cells, and we even tried different spatial omics technology that is labeling based and very non-destructive to the tissues. So if you look at these new next generation of spatial omics technology, such as Xenium or Akoya, as another method of staining. We could perform this spatial omics technology, and then choose the cell type that you want to isolate. And you could apply SLAX technology to isolate that cell and add a different omic modality. For example, if you perform Xenium, you get the transcriptomics data, and if you isolate these cells with SLAX technology, you can add like mass spec data to add that proteomic modality to that. So you would have that very good multi-omic approach in understanding the mechanism behind certain biology, or finding that different targets from different modality. You can find RNA targets. You can find the protein targets from the same biospecimen, so that you would have that multidimensional understanding of these different targets. Also, we recently published with uh, uh, Professor Sung Kwan's group in Seoul National University, um, in Nature Biomedical Engineering, where we uh, extracted a very good prognostic feature, a prognostic image feature, using graph deep learning. So you could use AI to find that image marker that is very relevant to determining the survival probability of different patients. And if you have that image biomarker, you can use SLAX to convert that image biomarker to a good molecular marker when you isolate these image features into PCR tubes and perform DNA sequencing or proteomics using mass spec, for example, and you would have that molecular explanation of that image, and you would have that molecular target to develop on. So we have a very collaborative network of a biomedical research center that is formed with our company and several groups in Seoul National University and Seoul National University Hospital, so that we could use this AI-based, machine learning-based feature extraction and connect that extracted part to the molecular diagnostics or molecular therapeutics part, so that we would have that AI-assisted um, uh, um, um, extraction of a important feature from certain diseases, including cancer, brain neurodegenerative diseases, many other inflammatory diseases, and autoimmune diseases, and so on. So we currently have a resolution of the 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 product that we produce has a resolution have what in of a 3 micrometer by 3 micrometer to 300 micrometer by 300 micrometer. So it could be a very dynamic range of region of interest or ROIs. If you look at it's a little small, so you might be able to difficult to see it, but it's a picture of isolating single E. coli from a mixture of different bacteria in a glass slide after gram staining. If you look at B, is a isolation of single chromosome after Giemsa banding and dispersion of different chromosomes, and you were we were able to isolate a single chromosome and perform PCR different omics technology so that you could have that extract out genetic feature from these single chromosome. And single cell, we were able to isolate in C after right Giemsa staining, and in D, very fibrous regions in breast cancer or very rigid regions, as you can see in E, in bone tissues. We could also apply SLACs to isolate different parts of the tissue. So very fibrous, very rigid tissues could be also be isolated, and also this kind of technology is very useful when it comes to isolating very rare the rarest cell population from a biological system, where it is below the limits of the resolution of isolating it when you're using dissociated samples using FACS technology or MAX technology. Um, if you have, for example, the rare population of cells that is below 0.01%, you will see these cells in our glass slides, so you will be able to isolate these cells. But if you're using FACS or Max machine, you would have to need a huge number of cells to input, so that you would have that rare cells to analyze. So various versatile use very you could have very high creative research using our technology, and this video shows a real time of isolating different cells. This was HER2 positive breast cancer tissue, where a pathologist pre-labeled a regions that could be a precursor or invasive population of breast cancer, so that this machine would automatically find those labeled places and isolate different parts of the tissue so that we could perform DNA sequencing and RNA sequencing. So basically, the philosophy of the SLAX technology is, if you have any kind of tissue that could be, or any kind of biospecimen that could be prepared on a glass slide, you can prepare it on our glass slide, which has very similar, almost the same physical property as the normal glass slide would have. So any kind of biospecimen that could go on top of our slide is a fair game to be used. And after these cells are isolated, the cells could be lysed so that the DNA, RNA, proteins inside these cells could be extracted out to perform omics technologies such as genomics, transcriptomics, and proteomics, so that you could have that omics information mapped back to the spatial information, so that we could have this spatial network of different cell interacting with each other, and you could have that very important, spatially or pathologically important targets of the cells, and you would have the deepest information that you could get from these cells, because you have that a very good quality of genetic molecules that are inside these cells. This was a case of 3D analysis of triple negative breast cancer tissue. And as you can see, we could map out very rarest subclones that are inside this triple negative breast cancer, so that we could match the genomic abnormalities to the subclones, the pathological information, so we could understand the tumor evolution and different genomic signatures of different subclones that are existing in one patient, so that this patient would have each and every genomic information for each and every subclones that the same patient has. And this also applies to liquid cancer, such as leukemia or myeloma. We could extract out single cell from these myeloma or leukemia samples, so that in this case was a leukemia sample. This leukemic patient had a very large population of leukemic cells, and we were able to isolate these cells in single cell level and sequence them, define their genomic abnormalities, and follow up on their MRD, minimal residual diseases, or even find the molecular target, so that this targets could be translated to different um, therapeutic strategies, such as ADC, antibody-based, or mRNA vaccines, if you do mRNA sequencing. Um, so we would like to position our technology as the world best target finding technology. We published this paper in 2024 in Molecular Cancer. Um, and because this is a spatial omics technology, we often compare our technology to the spatial um, space technologies, such as telescopes, Webb telescope, spacecraft, and so on. So if you look at these labeling based spatial omics technology as a technology where you can see the stars, how the stars are located in certain space, our technology is more like sending a space probe to those stars, so that you could dig out the deepest information from the star, so that you could have a very deep information from these cells. So as I mentioned earlier, these technologies, the telescope-like technologies, are more suitable for investigating the tumoral landscape or biological landscape of the tissue, where our technology and very similar technology called laser capture microdissection technology that uses ultraviolet microdissecting technology is more useful in exploring the regions of interest of the tissue or biospecimen. But the big difference between our technology with a similar technology called laser capture microdissection technology is that we use infrared pulse laser instead of ultraviolet beam laser. So we have much faster speed of isolation, and we have a very um, very milder impact to the genetic components, because of the wavelength of the laser that we use. So we have also published this decision tree, where if you want to consider detailed target analysis, you could use our technology. But if you want like more broad overview analysis of the tissue, you could use fish label or in situ barcoding based next generation spatial omics technology. But I, but we think that the most important thing about spatial omics is to find the target of the therapeutic or diagnostic targets. So we have been working with different diseases, and we have published a few papers, around 20 papers, to demonstrate the power of our technology. As you can see from this example of where we published in 2022 in Nature Communications. This was a triple negative breast cancer patient cohort, where people had received neoadjuvant chemotherapy, Taxol grade neoadjuvant chemotherapy, and but still had a tumor residing after the chemotherapy. So the surgeons decided uh, uh, su- to perform surgery on these patients, and we hypothesized that cancer stem cells would be still residing in this tumor, that could cause the chemo resistance of the chemotherapy that these patients were receiving. and we try to compare these cancer stem cells to cancer cells, so to find that target of that could specifically target these cancer stem cells. So because breast cancer has a pretty well-defined markers for breast cancer stem cells, we stain them with cancer stem cell biomarkers such as CD44, ALDH1, and um, we were able to define these very rare cell populations in tumor sections, and we isolated them with SLASH technology, and we were able to produce full-length RNA sequencing data, and we were able to get gene expressions, alternative splicing variations, BCR sequences from the B cells, TCR sequences from the T cells, and even single point RNA mutations from these transcripts that are in these cancer stem cell compared to uh, cancer cells. So as you can see on the right, we immediately found this GPX4 mutant transcripts that is causing um, to inhibit the ferroptosis mechanism in these cancer stem cells when they meet this neoadjuvant chemotherapy therapeutic substances. And we took 109 patients from TCGA and divided them with a group that contained our immediately found marker, and the group that didn't have our immediately found marker, and the group that had our immediately found marker had much higher survival probability and while the patient group that did not have our marker had much poorer survival probability. So we're taking this data with Seoul National University Hospital, and you're using patient-derived organoids to test if a ferroptosis inhibitor or a ferroptosis enhancer, such as Eristin or any kind of antibody-based drugs, can aid taxol-grade chemotherapy to basically cure these chemo-resistant breast cancer stem cell population, so that they could potentially be used as a companion diagnostic, companion therapeutics with the taxane grade chemotherapy to the triple negative breast cancer patients, which have a very relatively lower survival rate compared to other types of breast cancer. So we're trying to we got a research grant from Seoul National University Hospital, so we are working with the hospital to see if this immediately found marker could actually be a realistic drug therapeutic strategy for these patients. And this was acknowledged by a Nature Reviews Cancer right after 2 weeks after this Nature Communications paper was published. The editor from Nature Reviews Cancer invited us to write an article from the tools of the trade section, which the section that uh, introduced tools that are essential for making scientific discoveries, and that was very prestigious of us to publish this short article from Nature Reviews Cancer. And we also won a very prestigious award, such as the Scientist Award, a / Innovation Avenue Award, and we were able to get first place in the scientist innovation. So to sum up, we have developed this ultra fast and precise technology with our 10 years of research know-how, and we want to convert this as a cutting edge biomedical technology. And we are a university startup, although our company is 2 year old, a very young company. But we do have 10 years of research experience. The bulky machine on the left was the machine that we built as graduate students in a graduate school in Seoul National University, and we made this manufacturable last year in 2024. As you can see on the right, we now have the benchtop solution for this device, and it has the same aspect of the feature as the original SLAX technology had, the speed, the precision, and also different applicability of the technology. And we also have a device with the RNA amplification solution, as well as our slide solution, so that users could use them. And in 2 years, we were able to get different R&D funds and different awards to basically um, uh, market our product, our technology to different users uh, in around the world. And we currently are in collaboration with different applications and according to different indications, so that we would have a very high expandability, high applicability of our technology in different diseases. So now in Korea, we have three main sites where they bought our purchased our instrument. One is Seoul St. Mary's Hospital and Catholic University Department of Pharmacy, and International University, as well as Seoul National University, Seoul National University Hospital. And the US, we have installed our technology, our instrument in Mayo Clinic right now, and University of Michigan, Ann Arbor, in College of Engineering as well as College of Medicine, so that they could use our technology for finding drug targets and diagnostic targets. And in June of this year, we are installing 6 more installations in Eli Lilly, 2 sites in Eli Lilly, one in Broad Institute, MIT, one in Stanford, and one in UCLA, where our US site will be in the UCLA core facility. So this is a 10-year history of our development of our technology. We had 2 years of engineering, and we had almost 8 years of setting up the bioinformatics, biochemistry, AI, different clinical applications, scientific applications, and we started publishing from 2018, and we started up this company in 2022 to push forward to translate this technology to the real market. And I want to finish up with our vision slide. We had very large projects, whether it be funded by the government or private sector. We had the Human Genome Project, the Cancer Genome Anatomy Project, Cancer Genome Atlas, Human Cell Atlas, and many companies, as I showed you in the first slide. Companies like Illumina or Tennis Genomics had a very pivotal role of commercializing these academic innovations to industrial innovations. But I think currently is the era of spatial atlas. Many companies are trying to commercialize spatial technologies into the market so that people could ultimately have a solution, therapeutic solution or diagnostic solution, that could be a real world holy grail to the clinicians or scientists, to cure every diseases for everyone. So we want to play that pivotal role for spatial atlas era, where people could use the spatial information to find the real world target to basically translate them to uh, uh, therapeutic strategies like cancer vaccines, new antigens, anti-tumoral antibodies, diagnostic solutions, digital pathology solution. So as Larry Ellison, the CEO of Oracle, said, "This we can dream that one day AI assisted healthcare could realize a 2 day cure for or cancer every cancer patients, and we are envisioning that our technology could aid that to accelerate uh, this scientific or clinical." Studies to realize that dream and ultimately produce a very clinically highly relevant discoveries that these researchers and scientists can make. Yeah, so that's the presentation I prepared for today. And if you have any questions, I'll be more than happy to answer them. Thank you.